Good afternoon, everybody. I'd like to welcome our listeners. Thank you for joining us today to hear the webinar that we've put on to showcase Lexitas Reading Center, ONL, and Voxeleron. Before I open up and introduce the panel, I'd just like to ask members of the audience, should you have any questions during the webinar, please feel free to place them in the comments, the chat, and we will get to those during the course of our discussions. So I'd now like to ask our esteemed panelists to all introduce themselves. So Connie, if I could ask you to provide some background first of all.
Sure, Sally, and thank you so much for having me today. My name is Connie Chang. I'm the Chief Operating Officer of ONL Therapeutics. We are a clinical stage biopharma company focused on developing novel therapeutics to address the needs of ophthalmic diseases. Our development program has included clinical studies across 3 different indications, including geographic atrophy associated with dry AMD, also glaucoma, and the one that we'll be focusing on a little bit more today is an emergent acute condition called retinal detachment. We're really excited to be here and share what we've learned in our process of working alongside Lexitas. Looking forward to it.
Thanks, Connie. And over to you, Daniel.
Hi, thanks, Sally. My name is Daniel Russakoff. I'm the CTO of Voxeleron. We're a software company based in Austin, Texas, specializing in ophthalmic image analysis, and it's our iNebula and Orion software that were used for this clinical trial.
And I'm Sally Tucker. I'm the President of Global Services at Lexitas. We are an ophthalmology specialized clinical research organization, and we have an integrated reading center that we're extremely proud of. We partner with Voxeleron on a regular basis. They are our technology providers. And I am honored to be joined today by Connie, the COO of ONL, with whom we partnered on their clinical trial. And we're going to discuss that a little more today. So Connie, I know you touched upon the other assets, the other areas that you're focusing on. Perhaps do you give us a little bit on where you are with your asset, where you are on that journey in these different areas as the first point of topic for us to talk about today?
No, absolutely. So ONL's lead compound is ONL 1204. We have been working on this unique mechanism of action that inhibits the FAS receptor that's on the surface of retinal cells. FAS inhibits both the cell death and cell inflammatory processes in the various tissues of the eye. And so it actually has implications for our lead compound being a potential therapeutic choice in a variety of different diseases. So what I'd said before is geographic atrophy, glaucoma, and retinal detachment. The company was founded in 2011 out of technology from the University of Michigan. We're actually based in Ann Arbor, Michigan. Go Blue, for those of you who follow American football. And we've been very excited to be able to advance the clinical development program. And in fact, this week we will be closing a Series D round of funding later this week, which will allow us to continue the development. So that's where we are.
Fantastic. Thank you for that. And we were honored to partner with you on your retinal detachment program. Perhaps you could share with the audience the most important images that you collected within that program that we participated together on.
Well, maybe I'll also start, Sally, by just addressing the fact that we, went through a process of choosing our CRO partner. We ended up choosing Lexitas because of your expertise in ophthalmology, because of the relationships that you have with the retina clinics, and that proved to be a very good partnership. The primary endpoint for our study, and just to give a little bit of background, retinal detachment does have a standard of care surgery. So what happens is, all of a sudden in an emergent situation, your retina kind of starts to tear off, and it can cause, irreversible photoreceptor loss if not reattached quickly. But there usually is a little bit of a gap between the time you're diagnosed and the time that you can actually have that reattachment surgery, which varies where you are in the globe, quite frankly. So our primary endpoint was to look at vision in a variety of different ways, contrast sensitivity was one measure, best corrected visual acuity, another measure. And in terms of imaging, as you asked about, we did look at OCT, which is one of the gold standard imaging modalities for looking at the posterior segment. And for that, it was very helpful to partner with Lexitas and your reading center.
Great. And perhaps you could share a little bit about the study itself, what it was like with working with Lexitas during the course of the study, a bit about your recruitment and how that went. I think that would be interesting to share.
Yes, definitely. Recruitment and enrollment curves are paramount in terms of importance to sponsors, and there's probably a number of them on the call today. So the vital statistics of our study was that we had about 40 sites all in the US working together with us to recruit about 135 patients. And this is, again, an acute emergent condition. So we had to get those sites to be able to diagnose, consent, in the process of a patient coming in saying, oh my gosh, I've lost part of my vision, because we were studying that form of retinal detachment where you actually, it's macula off. So you're losing central vision. It can be quite scary for patients. And so our process of working with Lexitas actually started even as we were, finishing the protocol, putting the finishing touches on the protocol, all the way through to the last patient, last visit. We had a very good experience. We actually enrolled ahead of time by almost 6 months of what was expected, I think in large part due to the teamwork that was involved between us as the sponsor and Lexitas as our CRO, and also, quite frankly, the strong relationships that we had collectively with the various retina personnel and also, of course, the principal investigators.
Thank you. I'm now going to switch a bit to the software. As I mentioned before, we partner with Voxeleron. They are our technology providers, and we work with them with the iNebula and the Orion software. The iNebula software provides our database, and the one thing that is unique with the iNebula database is that it provides real-time access to the sponsors that work with us. And it would be great to hear your perspective on how that worked for you, the benefits of it from your side. So perhaps you could share a little bit about that.
There definitely is a benefit and a value with being able to pull up images in real time or almost real time to better dig into some of the results that we're seeing. Our phase 2 study was actually sponsor unmasked, and so we were following along quite carefully because we needed to understand the parameters around how our lead compound was performing. So for instance, if we have a data point from a site, it was very helpful to be able to tap into the real-time imaging and determine, hey, is a revisit needed? Is this something where we want to make sure that it was collected correctly? Can we see if this tracks with what we would have expected if we look at what the baseline lesion size was? So things like that, I think, were really helpful. And in speaking with our clinical team at ONL, they definitely found it was very helpful to be able to tap into that.
Great. And Daniel, as the developer, perhaps you could give from your perspective, how you see that this real-time access in the database can benefit users.
Sure. I can add to that, maybe just highlighting 2 specific points. One related to the access and one maybe to the analysis. But first, right. So I think my PhD is in computer vision. I think it's important to understand that the one of the problems here, and that's the interoperability problem in ophthalmology. So I started working in radiology, and they are light years ahead of ophthalmology, in terms of technology, because all their images come in a standard format. In this case, DICOM. So like a CT is a CT, regardless of whether it came from a GE or a Siemens machine. The same is not true for ophthalmology. Each device kind of has its own format, it seems. And Michael Chang, the director of the NEI, pointed this out, saying it's stifled innovation and it's holding back the field as a whole. So what we've done is we've taken the time to do what the manufacturers won't and standardize the image formats so that we can display them in real time and put it in one common platform. So, as Connie mentioned, having access to the data, so having solved that problem, we can give sponsors access to their data in real time. And as Connie mentioned, that's important to dig into the trends. We're also seeing anecdotally that trial recruitment speeds up as the site PIs themselves can see the progress of the trial and they become more invested in the outcome. We're also hearing it makes data and safety committee meetings a lot more efficient and effective, having access to all the data right there. To underscore all of this, if your reading center can't get you real-time access to view your data, chances are they're using the OEM software for their analyses. And that brings me to the point number 2, and that's sort of analysis. The OEM software is designed for a simpler clinical workflow. It can't handle complicated endpoints. It's not supposed to handle complicated endpoints at this point. It's not really designed for a clinical trial workflow, and I can give you, and actually in a lot of cases, it can't even handle the simple endpoints. I can give you 2 specific examples. The Heidelberg Spectralis OCT software doesn't let you recenter the segmentation of the fovea if it doesn't get it right the first time. So if you have a complex case and the fovea is wrong, you lose that visit entirely, or you have to accept that incorrect result. TopCon, similarly, they won't let you edit the layers, or they will let you edit the layers, which can work clinically, but they won't let you save those edits. So in a clinic, that might be okay to get your data point with the patient in front of you, but for a clinical trial, it's not acceptable, and you basically lose that visit too if the pathology is so great the segmentation is off. And in none of the cases of these cases can you get advanced endpoints like quantification of fluid, et cetera. So, by contrast, we have all the data in a single unified platform with the tools to perform all the advanced data analysis that you need and the power to edit them if necessary.
Great, thank you. And I think that brings us nicely to another aspect of the software, which is unique to us, which is the fact that it's agnostic, device agnostic, and you can use it across multiple different devices. So perhaps you could give us more information relating to that, Daniel, and the advantages that offers.
Well, I mean, I think it's as I said, the fact that ophthalmology is not like radiology, and every device has its own format. Either you have to commit, if you want to analyze things in one software platform, you either have to commit to one specific device, which limits the number of sites you use, and you have to commit to a limited number of endpoints. If you have device agnostic software that can read in any ophthalmic image and perform all the analyses in one spot, you're no longer limited to those, and it speeds up recruitment. It improves the results. You can make the trial much more effective and efficient.
Thank you. So Connie, I'd like to ask a direct question now, and it is, from your perspective, having used Lexitas Reading Center, what do you think were the key advantages of using our Reading Center over others?
There are a variety of things that lessons learned that we had, very positive lessons learned in working with Lexitas. I think one of the things, and this goes globally beyond just the reading center. So when we selected Lexitas, it really was around how do we feel about the partnership? Do we feel like we can have an open dialogue based on trust with the team? And that was really important to us because we know as a sponsor that best laid plans always encounter hiccups and bumps in the road. And so I will say that the ability to team with the Lexitas group on a number of different areas, including at the very get-go, finalizing the protocol, was really important to us. We also felt like, and I felt like on a personal level, Sally, I could reach out to you and we could say, hey, this is going really well. Let's do more of this. Or, hey, we're hitting a snag here. Maybe we need to rethink, where the resourcing is going, just so we can get ourselves, to the place where we need to be. I think the critical success factor for us was really being able to understand a little bit more about just how strong those partnerships with the retina clinics were on the Lexitas side, that we could ensure that the PIs were very, very engaged the whole process. To Daniel's other point about interoperability, I think that was part of getting the sites on line so quickly and get them really excited about enrolling patients was that they didn't have to worry about, do I have the right piece of equipment? We didn't have to have a lot of barriers to having the participation. And that was, I think, fundamentally helpful in establishing a strong positive relationship with the sites right at the get-go. I think all of that translates into what worked really well with us working with Lexitas and with the Reading Center in particular. And I can't emphasize enough, when you have enrollment faster than you expected, then you're going to get the study done on time, under budget, and you're going to be able to get more quickly to developing your product that will hopefully help those patients at the end of the road.
And that's what we all want. We want more treatments to patients as quickly as possible. We've touched upon the iNebula software and the Orion software, but I know, Daniel, that there are other tailor-made solutions that you can offer to sponsors. So perhaps we could touch upon that.
Sure. Yeah, this was a particularly interesting trial to work on because it exercised a lot of our unique capabilities. So for example, we use an area-based intersegment outer segment ratio metric that we customized for ONL with some feedback, some back and forth with them, and it's a metric you just wouldn't be able to get at other reading centers. And so in addition to the standard layer thickness measures, in this case for Spectralis and Cirrus images, we were able to work with ONL, offer OCT angiography quantification, fluid endpoints. All in all, it was a pretty complex trial, and those are the most fun. And I'll say, this was a single read trial, but we were also able, that was the requirement here, but we were also able to customize workflows. We are also able to customize workflows, depend on a trial specific basis. So double read with adjudication, whatever you can imagine, we can implement.
Excellent. And from a sponsor side, there's lots of discussion, a lot of excitement about biomarker identification. Connie, perhaps, you could provide your perspectives on those.
That it's very exciting to see how AI analysis is starting to make some strong inroads in ophthalmology in particular. The association between imaging and quantification of those imaging with AI and eventual visual function loss or gain is really important. And I used as an example geographic atrophy, where ellipsoid zone has already started to be recognized as a potential approvable primary outcome parameter. So I think it's important to always be pushing the envelope. As Daniel had mentioned, perhaps ophthalmology imaging was lagging a little bit behind general radiology, but I think it's definitely catching up, and that's going to be good in the long run for physicians and good in the long run for patients.
Excellent. Thank you for that. And Daniel, perhaps, you have a few words that you would like to add on the post-hoc analysis that you can offer sponsors and to look for biomarkers and so forth.
Yeah, absolutely. I mean, we're at an interesting point in the AI revolution. Certainly this last year has been all about what AI can do. But I think we're starting to see a little bit of slowing down of that hype. There's a lot of the things AI can't do are starting to get a little bit more publicity. So, for example, the Razor Bill trial had a lot of hype in the ophthalmology community about the power and promise of AI. But if you look it up on clinicaltrials.gov, the conclusion was basically it didn't help in that context. So I think I'll get to post-hoc analysis, but I think Phil Rosenfeld kind of said it best. Recently, what we're doing right now is really machine learning. It's not fully artificial intelligence, so we're not quite at the human level. Like I know there are a lot of people are predicting a world where clinical trials would be done entirely without reading centers. AI would do all the reading, but I think we're a ways away from that. But that said, if you're just using OEM software for your clinical trial endpoints, you've fallen behind the curve. There's more and more AI-based endpoints. They're going to become more and more prevalent. Just like as Connie said, as therapeutics get more advanced, these complement inhibitors, with respect to GA, I think they're going to have the same effect that anti-VEGF had for OCT, just sort of, making it ubiquitous. And to that end, as I said, we have in-house a number of research directions involving advanced endpoints. We have GA segmentation, we have fluid quantification, we have quantification of photoreceptor loss in-house. And whether it's part of a post-hoc analysis or part of a exploratory endpoint, we're ready to bring these technologies to bear.
Thank you for that, Daniel. I do have a question from the audience, actually, for you, Daniel. Can you describe the workflow for using the iNebula software? Does the site install special software on its existing devices, or does the site need a new device that is compatible with the Voxeloron software?
Nope, iNebula is entirely browser-based, or iNebula lives in the cloud, so it's entirely browser-based. All site needs is a login. So they log in. We would do do an onboarding session with the sponsor, where we get the site accounts, the visit schedule, the CRF, the analytes involved. Once we enter those and create the accounts, all the site needs to do is go to the iNebula.com or lexitas.inebula.com in this case, and drag and drop the images for that visit. If you want a demo, if anyone wants a demo, please, I think our emails are publicized out here. Please don't hesitate to get in touch. We'd be happy to show you.
Absolutely. Thank you for that. And it's going to move me now to my last question, and it's for you, Connie. So do you have any lessons learned that you could share with the audience about this program that you'll carry forward to your other trials, or that you'd like to share with the other sponsors listening in today as they're evaluating reader center options?
I think it starts with the sponsor having a very good sense of what they're going to need to be successful. And it sounds like a very obvious statement, but in the context of a small biotech company where there's a lot being covered by relatively few people at the company, prioritization is very, very important. And so if you know what it's going to take to be most successful with your trial, and you can articulate that even as you're looking for the right reading center partner, even as you're looking for the right CRO partner, that's really important, because you're going to get the best kind of response to your RFP process if that's where you are. So taking some time at the front end is really important. The second thing I will say is the earlier you can involve your partner, the better. So I had mentioned that we worked alongside Lexitas with protocol finalization. I think that was, in retrospect, a great way for us to not only finalize the protocol in a team-based fashion, but also be able to pressure test choices that we could make about entry criteria, exclusion criteria. Are there downstream impacts to how easily or more difficult it will be for a site to screen patients if we include these parameters? Should we put them in a nice to have bucket? All of those things, I think, can result in adding grind to the process or facilitating the process from the site. And it actually also helps in building that trustful relationship between sponsor, CRO, and sites that is, again, can make or break the enrollment curve in any clinical trial. I think last but not least, I think we had built in some very good regular touch points, both at the study team level, but then also had very transparent escalation processes and people that folks could turn to, both on the Lexitas side and the sponsor side, in case things, needed another layer of conversation, which isn't always a bad thing. And I think having those touch points predefined was really helpful. So all in all, a good experience for us.
Great. Well, thank you, Connie. Thank you, Daniel. We appreciate you both. We appreciate your partnership. Connie, thank you for collaborating with us on your important study. It was a pleasure to work alongside you to complete the trial. And thank you both for your time today.
Thanks for having me.
It was a pleasure.
Yeah, thank you.
Pleasure, Daniel.